Just want to thank David for basically setting the stage, sort of from a grander climactic picture for just my portion of the presentation, which is really looking at sort of a case study and how research trials that we've done with our collaborative partners, grape growers and wineries here in California. I'm looking at sort of building vineyard resilience through a soil management, sort of soil microbial management product. So here's the agenda, really, for today. This is sort of going to start off with an overview of heat waves. I'm just going to very briefly discuss this because David sort of already covered sort of most of what I would need to say here. And then we'll sort of look at the research of heat wave resilience and how that correlates to the soil microbiome, because that might not be sort of as common sense as one might think, but there is a correlation there. And then we'll sort of launch into the meat and potatoes of at least my portion of the presentation, really looking at the BluVite technology and how we've used certain protocols using infrared thermography and then ground truthing that or validating that with actual plant water stress measurements, and then trying to make some correlative comparisons looking at berry dehydration and doing some seasonal yield comparisons. And then we'll have a brief summary at the end. So this map was actually created using the Goddard Earth Observing System, the GEOS model. This was taken on September 6th. This image basically represents temperatures roughly about 2 meters above the ground, which is about 6.5 feet. And the darkest colors, the darkest red colors, are areas where the model is measuring temperatures above 45 degrees Celsius, which is about 113 degrees Fahrenheit. And this is actually the same day where record high temperatures were measured across California. Paso Robles, for example, measured a high temperature of 111 degrees on this day. I believe Los Angeles County also measured a record temperature in Woodland Hills of 120 or 121 degrees Fahrenheit. So this is sort of gives you an idea of what we're up against in terms of heat waves in California. So moving on to a study done by Caltech University, or I guess Caltech in California, was basically published in June of 2020. And this particular study looked at data collected for inland urban areas of California from 1950 to 2020. And the three graphs basically from left to right, from the orange graph to the blue graph to the red graph, basically the first one is visualizing the number of unique heat wave events per year. The second graph is looking at the sort of duration of these events in days. And the third graph is looking at the intensity of these events in Celsius. And the heat wave intensity is actually calculated, at least for this study, is calculated by subtracting the average heat wave temperature, basically subtracting that from the maximum temperature of that heat wave. And so this study summarized that these heat waves are occurring more frequently in terms of heat waves per heat wave events per year. They're lasting longer in terms of duration events in days, and they're becoming more intense as well. The study also stated that coastal areas are less affected when compared to inland urban areas, and this is because there is some coastal influence. But as David had mentioned, ocean temperatures, Pacific Ocean temperatures are increasing over time. And so coastal areas are not unaffected. We have actually seen a threefold increase in the frequency of heat waves in the last 20 years. And, if anyone who experienced the summer in California will attest to this. So now moving on, this is essentially looking at sort of soil microbiome in reference to heat waves. And this graphic on the right-hand side is essentially looking at a heat wave in relation to this soil microbial community, this microbial consortia, this soil microbiome, whatever your preferred terminology is. And this is really looking at in relation of sort of the composition or the biodiversity of this consortia and sort of the metabolic activity in relation to the rooting zone, the rhizosphere. plant growth, and in our case, vine growth. And this is sort of the interaction, studying the interaction at the microbial level. And then also looking at heat waves in terms of ecosystem level, how the biological component interacts with the physical and chemical properties of that soil. And basically looking at sort of current research in the sort of with the soil microbial community at the center of this, there really isn't too much research out there. It's sort of a fringe science at the moment. But I do imagine that with the more frequent heat waves, there's going to be a growing body of research labs that are starting to dive into this to look at if the soil microbiome can improve grapevine resilience. The effects of high heat on grapevines have been very well documented, especially in terms of yield and fruit quality. High temperatures do have a negative effect, especially in terms of grapevine physiology, and this ultimately has huge implications for reproductive development, which is the grapevine clusters, which is what we use to make wine, and has negative impacts on fruit quality. This study, where I got the graphic from, essentially looks at microbial recovery strategies in particular to see, especially in regards to vineyards or agro-ecosystems in general, if certain microbial recovery strategies can help to make the system more resilient towards these heat wave conditions, which ultimately are going to become more and more common. And so this is sort of the inspiration for these trials here in California. This technology is essentially a microbial recovery strategy that is not introducing an inoculum. It's stimulating the native microbial consortia, that native microbiome. And so through this, this microbial community interacts with the rooting zone, the rhizosphere, increasing that rooting biomass and making nutrients more plant available, essentially allowing the grapevine to better utilize its resources. This actually makes for a grapevine, more resilient grapevine, and I'm going to show that to you in the following slides through the case study. And yeah, again, these trials in California were really to work with growers to see if we could discover any tools that would be helpful to withstand, into the future seasons that are similar to what we experienced in 2020. And so this is sort of just kind of jumping straight into the experimental design and the experiment itself. I'm just kind of going to first cover sort of an overview of that experimental design. It's kind of important to sort of place this in context to sort of show you how we design these trials. So vineyard selection is probably the most important part of this of these trials, because you want to make sure that sort of the natural properties of the vineyard, soil type, soil physical and chemical properties of the vineyard are uniform across that vineyard, where you're going to put your control and your treatments. And then you also want to take water availability into consideration too as well, and vigor plays a huge part of that. So you have to use NDVI images and look at vigor in the vineyard and pick something that's more uniform across that vineyard. And then there's characteristics that humans ultimately make the decision on, which is, clonal type and rootstock and vine spacing and sort of those characteristics. And you want to make sure that everything is the same across that entire vineyard. And then I'm not going to go into detail as to what the BluVite product is, but the important thing to note is that it's applied in the vine row. There's two products, I believe, that are available in California. One being one that's fertigated and one that's applied directly to the soil. But the important part here is that it's applied in the vine row. And then the vine row is essentially where most of the active roots are going to be. And so this is something important to mention too as well. So irrigation management, canopy management, floor management, nutrient management of these vineyards across the whole vineyards are kept exactly the same for these trials, so that the only variable that we have is this technology. And so, and this is an important thing to note, because we want to have data that is meaningful to the growers and to our collaborators. And so everything is measured in triplicates, so we get better statistical significance. So I think that's important to note. And this is how most trials are done, but it's just something that I need to verbalize before we move forward. So this is the experiment here. From this image, you can see that the thermal camera mounts on top, plugs in directly into an iPhone or Android phone. And on the tailgate, the right hand of the tailgate, you can see the calibration plate or calibration placard. It has two pieces of felt on it. And essentially one of the pieces of felt is left dry, which it would be the warmest a leaf could be, and one of them is sprayed down with water to designate the wet temperature, which is the coolest a leaf can be. And it produces an image like this. So you can see the calibration plate, you can see the two pieces of felt. The hottest the canopy temperature can be is about 120 degrees Fahrenheit for this image, and the coolest it can be is about 87 degrees. And this calibration is really essential to make any sort of correlation or comparisons between treatment and control. And hopefully you'll see that this is actually a pretty good tool to use in your vineyard to study comparative management practices. And in this case, we're using a soil microbial product in that comparison, but this can be used across many different management practices. The experimental protocol was taken from Belfiore et al., and the study was published in 2019. And so you can actually use these thermal images, this infrared thermography, to estimate crop water stress, and also, crop water stress can be used to make irrigation decisions, and then also it can be correlated to stomatical conductance, which I'll explain later on in the presentation. And then again, we wanted to validate this, just like in the study, we wanted to validate our comparisons with on-the-ground plant water stress measurements. We mostly took leaf water potential measurements, but the example that I show in this presentation is Predawn stem water potential. All of these images and the crop water stress index and the stomatical conductance to dehydration and sort of yield comparisons. So this is also part of this experiment that we ran this year in 2020. So this is the first of the graphs. I didn't want to show all of the data, so I just decided to show the deltas here. And you have the 8 different AVAs on top, all in California. You have 2 in the Central Valley. In the Lodi AVA, you've got 2 in the Paso Robles area in San Luis Obispo County. You've got 2 in Lake County, the Red Hills AVA and Clear Lake, and then you have 1 in Sonoma County, Alexander Valley, and then you have 1 in Napa County. And so basically what you're seeing here is the average reduction in canopy temperature. And this is essentially using the data export, each pixel in the image that I showed you in the previous slide. Each pixel is a temperature. And so you can get average canopy temperatures across huge areas of the vineyard just by taking a picture. And so from this data set, you can see that on average, we saw a decrease of about 2.2 degrees Fahrenheit in canopy temperatures between the treated area and the control, which has pretty big implications, especially for how frequent and intense heat waves were this year. Even a reduction in a couple degrees Fahrenheit makes a big difference. And so this is essentially, you can see the formula. This is crop water stress index. The formula is in the right-hand corner here. These are the exact same vineyard sites, exact same AVAs. And this is a comparison again between the treated and the control. And using this formula, we're able to get indices or values, and these are the deltas between those values. And it's given in a percentage. And so you can see approximately 30 degrees less or average decrease in the crop water stress index between vineyards treated with BluVite compared to the control. And this actually aligns fairly well with the changes in canopy temperature that we picked up as well. And then another indices that's calculated from these images is the stomatical conductance index. And I'm sure most people are familiar with this, but for those who aren't, stomatical conductance is essentially related to the opening and closing of the stomata on the leaves of the grapevine. And so a low stomatical conductance reading really relates to the closure of the stomata and leads to a reduction in plant transpiration or vine transpiration and ultimately photosynthesis. And so if you have a low reading, this ultimately results in, in sort of water stress and higher canopy temperatures due to a reduction in the sap flow. And so ultimately, we did the same thing, looking at the treated adversely control and all these different AVAs. And given it as a percentage, we saw that there was actually a 42% increase in the average smell conductance index between the two treatments. And again, this has pretty huge implications for sort of the productivity of a vineyard across the season. And then, like I said, we wanted to sort of root this in measurements actually taken in the field. And so we took that measurement from the Rutherford AVA, which was in the past 3 slides. Here's the summary here. In the first graph, we saw that had a decrease in 2 degrees Fahrenheit in sort of canopy temperature. There was roughly a 6% decrease in crop water stress index and a 9% increase in smell conductance index. And so we tried to find measurements. The grower collaborator was collecting Predawn stem water potentials. We tried to get the data set that was as closely timed to the actual thermal imaging that we collected. And so what we found here, you know, sort of what it validates what we found in the infrared thermography. We saw essentially nearly a 1 bar decrease in the Predawn stem water potentials. Ultimately, with the lower reading, meaning less stress. And in block 1 and block 2, those two blocks had statistically significant differences. Whereas block 3, we did have a difference, but it was not statistically significant. But yeah, again, nearly 1 bar decrease. Anything really over 6 bar in Predawn stem water potentials is approaching stress. Anything, you know, in block 2, you almost get up to 7 bars. I guess it's negative bars, really. So significantly less stress, and this aligns really with what we found in the thermal imaging. So this really does appear to be an effective way to estimate plant water stress in the vineyard. And so we've got many collaborators who collected quite a bit of data, and so we're still really getting through the data. The season hasn't ended, really, and so we're really trying to get through the analyses and really kind of correlating it to the rest of the trial examples. But I just wanted to show you one example because I think this is a good example to show that this thermal camera can actually truly predict sort of on the ground validation of these water stress measurements. Another thing that I wanted to look at and to show you here today, and this is ongoing too as well, but we did see quite a bit of dehydration. You know, high heat is obviously not good for the integrity of the berry, the berries themselves, the clusters. And so dehydration was very, very common throughout our trials. And really, this is, you know, we hadn't seen this severe of dehydration in vineyards in the past. And I believe this is due to the increases in heat and water stress that we saw this year. And, you know, berries are really, the rate and amount of water evaporated by the berry is really determined by the temperature and relative humidity in that vineyard. And, you know, it's sort of intuitive to think that berries, you know, they will inevitably dehydrate faster in a hotter and drier condition. And so if we're measuring a reduction in canopy temperatures, then there's likely going to be the same thing occurring in the fruiting zone, which is ultimately part of the canopy, just the lower part. And what we found here, and what I'm going to show you in the next slide, is that we saw generally an overall reduction in the dehydration in the treated areas of the vineyard. And we measured this by looking at sort of yields, average cluster weights. So this is what this graph is showing precisely here. This is an example from Napa Valley AVA Cabernet Sauvignon trial. And we've got 3 samples here. The first one being late August, the second one being late September, and then the third one being just before harvest in mid-October. And so you can see here general decrease in yields for I mean, there's obviously a natural process that occurs throughout the summer through ripening, especially something that's hanging on the vine until mid-October. You get a natural decrease in yield, but in the treated area, there was basically 2% higher yield performance under these heat stress conditions. I'm not sure exactly how many heat waves were between late August and mid-October, But that one chart that I showed you in the very beginning was in early September. And so this vineyard, undoubtedly, those clusters went through some very, very high heat this year, especially on the valley floor in Napa. And so, again, like I said, we really think that these improvements in yield could be correlated to overall cooler canopy temperatures during these heat waves, and potentially due to maintenance of higher stomatal conductance measurements to keep these canopies cooler. And again, we've got so much data to look at. So this is an ongoing analyses that we're going to have to work with clients and our collaborators, growers and wineries, to sort of look at our findings from this year and move forward with tools that they can use to sort of reduce degradation of quality in these vineyards in terms of yield and shrivel. And so just a quick summary here. I think this year, and especially in terms of the larger climatic perspective in California, is actually, it's probably a good predictor in terms of the sort of types of growing seasons to expect in the future, especially in terms of heat waves. And so, this year really, not ideally, but it did present us a research opportunity. Just due to the frequency and the magnitude of these heat waves, our vineyard collaborators were really interested in sort of collecting this data, and especially when using technology like BluVite, which ultimately stimulates this root microbial interaction, because these are some of the worst conditions we've seen so far. And so this really was a great research opportunity for us and our clients. And sort of next steps, at least from a data perspective, we are seeing decreases in canopy temperatures, and then plant water stress measurements. We're also seeing these same decreases and increases in stomatal conductance. And so the next steps is really to sort of bring our collaborators and our growers that we're working with, and wineries that we're working with, and putting that onto the stage at the university level to further validate these findings. And I think that the next couple of years we're going to have some pretty exciting things to bring to growers to show what kind of tools they can use in the vineyard. And then so conclusions here, essentially, based on the research that I showed to you today, I hope that it became clear that BluVite can be a tool, one of the many tools. There's shade cloth, there's certain canopy management ways of managing the canopy that you can do to sort of combat these heat waves, and even designing your vineyards different. But the BluVite can be a tool in that toolkit, essentially to improve the vineyard resilience in a natural way. And so that's sort of the take-home conclusion here, is hopefully that BluVite can be seen as a tool to actually combat these heat events. Hey, thank you for attending this presentation. Again, here's my information. If you like, I'm a bit of a soil nerd, so I like talking about soil, so feel free to shoot me an email or give me a call. I'd love to talk anytime. And if you have any questions about the product that we used for these case studies, please email the general BluVite email address, and someone will be there to help answer your questions. Thank you.